Hi, my name is Eve Robinson, and I'm at Humboldt State University in California working with the U.S. Fish and Wildlife Service. I'm going to share with you today a model predicting the density of a parasite, Ceratonova Shasta, that impacts juvenile salmonids. And this work is done in collaboration with my co-authors at the U.S. Fish and Wildlife Office in Arcata, Oregon State University, and the USGS Columbia River Research Laboratory. We'll briefly talk about fish pathogens and management tools for disease mitigation, and then about the waterborne density of sea Shasta in particular, components of the model that we built to predict parasite density, our model results, and next steps. Parasites co-evolve with their hosts, and they form this dynamic equilibrium. When you significantly alter the environmental conditions, this can favor the parasite and cause the equilibrium to shift out of balance, which is then expressed as elevated infection levels in hosts that occur sort of beyond the background levels. These are a few of the pathogens that can affect salmonids. Those commonly known as ick and columnaris were attributed to the 2002 fish kill on the Klamath River that resulted in between about 35,000 or as many as 65,000 fish dying over the course of a few weeks. More recently, sea Shasta has been the focus of research and management in the Klamath because of its effects on the survival rates of juvenile Chinook and coho salmon. The Klamath Basin is located in southern Oregon and northern California, and it runs from Upper Klamath Lake down to where it meets the Pacific Ocean, about 55 miles north of Humboldt State University. There are 6 dams on the main stem. Iron Gate Dam is the furthest downstream, and it limits the passage of anadromous fishes. A hatchery at Iron Gate produces smolts to really mitigate for that habitat loss. For those of you who are following along in the news, the lower 4 dams are the ones that are slated for removal. Sea Shasta is a myxozoan parasite, so it alternates between 2 hosts and 2 waterborne stages. Actinospores are released into the water from benthic annelids where they encounter fish hosts. And infected fish then release myxospores that are consumed by the annelids, develop within these hosts before being expelled back into the water, and so on. The fish hosts that actinospores encounter really depends on the season. So in the spring, this is predominantly juveniles or smolts that are in the river, and in the fall, it's the adult fish that are returning to spawn. Within the fish, actinospores proliferate in the intestine, and they cause enteronecrosis, or gut rot, hemorrhaging, and lesions. Infections also leave fish susceptible to other pathogens, predation, and it compromises their osmoregulatory system that's necessary for transitioning into saltwater. Infection rates in Chinook salmon, for example, can be as high as 60% in the wild populations and over 80% in hatchery origin fish. So for the past 15 and more years, an extensive monitoring program has collected data on sea Shasta thanks to many people in the U.S. Fish and Wildlife Service, the Karuk Natural Resources Department, Yurok Tribal Fisheries Program, California Nevada Fish Health Lab, and Oregon State University. And these data inform the biological opinion that's released by NOAA that assesses the impacts of dam operation on species listed under the ESA, or the Endangered Species Act. One of the management approaches for sea Shasta targets the annelid hosts by using flushing flows in the spring that scour them from their habitat to then reduce their abundance before they release actinospores. Outmigrating salmon in the Klamath pass through what we call an infectious zone, or an area of really elevated spore densities. So the dam plays an important role in the location of this zone because it effectively concentrates spawning. Myxospores that are then released from infected adults or their carcasses in the fall are dispersed downriver, and they're deposited in areas that then overlap with annelid habitat and thus sort of create a source of spores in the spring. So our objective was to develop a model of sea Shasta density to predict some of these key characteristics of seasonal spore density patterns that relate to disease risk for outmigrating juvenile salmon. This model is also needed to evaluate management actions, such as directly assessing the impact of proposed flow regimes in reducing spore density, or indirectly as a submodel for existing management tools that estimate disease-caused mortality of juveniles. Our model needed to capture these 3 main characteristics of spore density shown here. So you can see A, the timing of the initial release or onset of spores from the annelid host, B is the seasonal pattern of spore densities, and C is the peak magnitude, which is different year to year. Model development was really underpinned by data from long-term monitoring studies on the Klamath, which really explore the biological and environmental components that affect spore density. Here are some of the biological factors that are hypothesized to impact the disease life cycle that might provide relationships useful in predicting spore density. So I provided a wee life cycle here for reference. In annelid hosts, we looked at the population density and prevalence of infection. In the salmonid hosts, we looked at the prevalence of infection in hatchery origin smolts from iron gate and the number of adult spawners. We also looked at peak spores in both spring and fall and the release date, whether it was early or later in the migration season. So for environmental factors, we explored river discharge regulated by Iron Gate Dam and the occurrence of flushing flows, which are defined as over 171 cubic meters per second in the spring. And we looked at water temperature, both the cumulative and peak temps, and the accumulation of thermal units or degree days. We also included annual measures of water year, such as precipitation and reservoir level in the Klamath River Basin. So I'll talk about each of the components or modules that address each different spore density characteristic and then discuss the final spore model. So first, to capture the onset of spores, we used a process-based biophysical degree day model that combines both biological and environmental components. Working backwards, we know adult spawners are the source of myxospores that affect the annelids. So we started the model based on the timing of the run, or when the fish enter the system, in the fall of the year before. We also know that once infected, spore development in the annelids is temperature dependent, and so we used a degree day model to estimate when spores would reach maturity. However, even once they're mature, spore release can be delayed due to low temperatures or the occurrence of a flushing flow. So our results predicting the week of the onset of sea Shasta is on the y-axis compared to the observed week on the x-axis, and they're labeled by year. 2011 over here was a year in which the spore densities remained near zero the entire season, or a very low disease risk. In general, the biophysical degree day model closely matched observations of initial spore detection from our monitoring data. To describe the seasonal pattern of spores, a curvilinear thermal response between sea Shasta spores and water temperature was fit with a quadratic curve for selected years. And this really just excluded years in which flushing flow events occurred as water temperature and flow are linked, and years in which the spore densities were extremely low. And then this module predicts the normalized spore density based off of water temperature. Finally, we predicted the peak spore density for each year, building upon a published linear relationship. So the hatchery releases approximately 5 million smolts each year upstream of the infectious zone, and while timed specifically to avoid interactions and competition with natural origin fish, it often means that it happens later in the spring that coincides with high levels of sea Shasta. So the black lines are spore monitoring, and the gray windows represent the timing of when hatchery release occurred. Having more fish hosts available when spores are high may seed the system for the following year. And we found an association with the prevalence of infection in hatchery origin juveniles, or hatchery POI, from the previous spring, and we used this relationship to predict peak spores. But we also wanted to evaluate some of those other biological and environmental covariates that we discussed earlier to potentially improve the prediction of peak spore density. So to avoid the risk of overfitting, we limited the number of selected variables to those supported by ecological hypotheses. We tested the improvement in model fit by adding each selected variable against the model containing only the hatchery POI. And we did this using likelihood ratio tests and found a strong evidence that hatchery POI and the occurrence of flushing flows were associated with the spring peak of spore densities. So our spore density model combines modules A, B, and C. I'm showing the predicted spores per liter in red, and monitoring data is shown in the black markers. Our predictive model successfully captured the onset of spore release, the overall seasonal pattern, and interannual variation. Importantly, it closely matched observed values, particularly for years in which density and therefore the disease risk is really high. As with any regression model, predicting the highest and lowest values is most difficult. However, this model predicts the variation with enough contrast to inform management decisions. So modeling parasite density requires an understanding of the dominant drivers of the parasite's complex life cycle that relies on, in this case, a wealth of monitoring data. The model presented here builds on these empirically derived relationships and includes both mechanistic and statistical relationships. Importantly, we included variables that are sensitive to management decisions, such as hatchery releases and the occurrence of flushing flows. This model is useful for simulating both disease risk for juvenile salmonids traveling through the infectious zone during the spring, but it can also be used into existing population dynamics models. And I'm next interested in applying this model to explore the impacts of disease under scenarios of climate change and dam removal. Again, thank you to my co-authors, and please post your questions. I'm always happy to talk about this work, and I look forward to hearing from you.